Hello everyone. Welcome to Module 15 of the GPU Teaching Kit. In this lecture, we're going to cover non-Cartesian MRI reconstruction application. The objective of this lecture is for students to learn how to apply the parallel programming techniques we have learned in this course so far to an application. There are four skills covered in this lecture. The first one is how we can determine the parallelism structure that we should be using to parallelize the application. The second one is how we can transform some of the loops so that they can become effective bases for CUDA kernels. And the third one is how we can adjust the memory layout of some of that data so that we can achieve a high memory access efficiency. And the fourth one is how we can validate the execution result against some application level requirements so that we know that the parallel kernels are producing the right kind of results. In the next few slides, we will provide some background information for MRI scan and image reconstruction. MRI, or magnetic resonance imaging, is a technique invented by Paul Lauterbach, and which uses the magnetic field pulses to measure the presence of different types of atoms in the object or a tissue. And the top formula in the slide shows the reconstruction of a object image or tissue image based on the K-space samples. M on the left-hand side is the reconstructed image that is a function of the space. And then the right-hand side are the formula for using the samples. And S is the K-space sample that the machine takes. W is the weight that we will be applying to the samples. And these weights are used to emphasize or de-emphasize the samples according to their density in the K-space. And then E is a basis function that we will be using essentially for the frequency domain to time domain conversion. The left-hand side of the slide shows a traditional approach to MRI scanning and image reconstruction. The MRI machine used in these traditional approaches will sample the data uniformly in the K-space. And we showed that these sample points as uniformly distributed samples in the Kx and Ky space. With such uniform distribution, the W will be of uniform value across all the samples. Therefore, the W can be taken out of the summation. Furthermore, the basis functions will also have equally spaced exponents. Therefore, it takes the form of very fast Fourier transform. So we can essentially create a reconstruction using the fast Fourier transform, which is a very inexpensive computational method. While the traditional MRI approach based on uniform samples in the Cartesian space is computationally efficient, they actually have quite a bit of deficiencies for medical applications. The most important deficiency is that they oftentimes do not provide enough resolution or fidelity in the areas that matter. So that's why there have been a lot of proposals for much more sophisticated scanning patterns or data sampling patterns to improve the quality and outcome of the reconstructed image. So here we show one of these more innovative, sophisticated scan patterns called spiral scan. And such scan gives the center of the K-space much more samples than the outskirt. So the one approach that we can use to reconstruct the image from such more sophisticated scan is to systematically transform the scan data into uniformly distributed Cartesian space samples. So this is called the gridding method. And we see that the spiral scan is illustrated as orange dots in the middle of the slide. And then all the Cartesian data samples that we would like to generate are illustrated as blue dots. So the idea is to perform a computation that would contribute from the spiral data samples into the grid samples according to the physical distance and so on. So this is a method that generates a uniform Cartesian scan data sample so that we can use the FFT approach to reconstruct the image. While this is a viable approach, it does have some shortcomings. One of the shortcomings is that when we do the gridding process, we may introduce distortions, and also the gridding approach does not allow us to be able to compensate for some of the artifacts that can be experienced in the scan process. Because of the deficiencies of gridding methods, it is actually more desirable from the application's point of view to reconstruct a MRI image directly from the spiral scan data. So on the right-hand side, we show such approach. We would use a more general formulation of the Fourier transform rather than the fast Fourier transform to reconstruct the image, and we use an iterative method to essentially adjust the image estimation based on a criteria to minimize an error such as, say, the noise in the image. And we use this method to estimate the image and minimize the error so that the image pixel values are of meaningful, the meaningful values, and we can use those values for diagnosis purposes. The more accurate pixel values in the reconstructed image enable some valuable medical applications. And this slide shows one such valuable application. The work is done at the University of Illinois at Chicago Medical Center. And Keith Thorburn's team used the MRI scan to map out the sodium concentration in the human brain cells. And sodium is a highly regulated substance in the human tissues. Any significant shift in sodium concentration signals potential cell death. So by scanning the sodium concentration map in the brain tissue and observe the shifting of these levels over time can tell us the trend of tissue evolution in the human cell brain. For example, during a stroke, if we monitor the sodium map development, we can see which part of the brain is going to have cell death due to stroke. And this allows us to pick the right kind of the treatment in a stroke process. And also for brain tumor treatment, we can use the sodium map evolution to tell if the brain tumor cells are going through the process of cell death, so that we can detect the efficacy of treatment before we see any anatomic change in terms of tumor shrinkage. And we can actually predict the tumor shrinkage by observing the shift of the sodium levels in the tumor cells. Now that we understand the motivation and basic principles of the non-Cartesian MRI reconstruction, we can now look into the details of its computation methods. Here we show the formulation of the computation method for reconstructing a non-Cartesian MRI scan. The middle of the slide shows the equation, which is the basis of the computation method. Rho is the image that we're trying to reconstruct. And on the right-hand side, we have FHD, which is based on the acquired data. And we have an operator, F Hermitian F time plus lambda W Hermitian W. And we're going to call this operator Q. And this operator can be pre-computed based on the machine characteristics and the scanning environment. So this operator also models some of the artifacts and the potential distortion that we have because of the environment. But the formulation is such that the reconstructed image, when applied by the Q operator, will be equal to FHD. So when we acquire data, we show the process on the right-hand side. We can compute FHD based on the acquired data. And then we use the pre-computed Q operator and perform a iterative solution to the equation. So we essentially do a guess of the value of rho, apply the Q operator to it, and then see if it is close to the FHD value that we computed based on the acquired data. And we iterate until the value of rho converges to the values that give us a close enough FHD value when we apply the Q operator. This is the computational method, and we're going to need to compute FHD for every piece of acquired data for every image that we construct. And so this is going to be the most time-consuming part, and we're going to be focusing on the computation of a F Hermitian D. Here we show the source code for the Q operator and the FHD. It turns out that the code has almost identical computational structure. The outermost loop iterates through M, which is the samples, and the inner loop iterates through N, which are the pixels. So we reconstruct. So if we look at both codes have the same two-level code structure, and they also have very similar, computational statements inside the innermost loop and also in the outer loop. So we are going to be focusing on FHD since it's a bit more interesting computation that will need to be performed on every acquired data in the process. This concludes module 15.1 of the GPU teaching kit. Thank you.